We're now here to go over chapter 14, the brain and cranial nerves. I know a lot of us will be very frustrated with the amount of contents that are in this chapter, but I'm sure this isn't bad actually. So long as we know how to study, this chapter is pretty doable. So I just wanted to know this chapter primarily focuses on the brain and cranial nerves, and we're going to focus more on the structure of the brain and how different regions and different parts of the brain actually work together to create some certain response to our environment. So before I actually move on, we can actually talk about several different things. I'm just going to skip a few slides right here. But before we start, it is very important that how your brain is actually structuralized to allow us to survive in the environment. So our brain actually formed and is structuralized and very localized in a certain way, such as environment. When we live in the environment, it always something happens, right? Such as um, your temperature changes out of nowhere. It gets hot, cold, something frustrating, something very stressful, some kind of stressful events, right? So something actually happens in our life, in our environment. So our brain was actually structuralized and developed into a organ or system that actually can counteract those stimulus with proper and necessary response. So our brain doesn't like to technically just bring random responses, but actually brain is very smart. Our brain is actually very efficient in providing necessary and proper response. So it actually differs in some emotions. So emotions can bring different response that we technically supposed to have, but such stressful events, such frustrating event actually can bring different kind of emotional response. So this is rather than having such as stimulus response. For example, you touch something very hot, and your reflex is to draw your hands out of that region. Such as those are stimulus response, but stress response are involved in actually emotional response. So those are actually a little bit different, but generalized concept is that our brain actually structuralized and localized so that we are able to respond to certain stimulus from the environment. So that is the main concept. And how our brain responds to it and how our brain is structured, that's what we're about to find out. So just make sure you guys understand what exactly our brain is doing. It's just receiving the stimulus, integrating stimulus, whether to respond or not. And based on the integration, our brain is able to provide feedback. So that is the basic mechanisms. So that's what we're going to talk about, like such as sensory, your visual, your optics, your olfactory, your smelling, your auditory, your visceral sensation, such as those, your pains, touch. Those are actually the stimulus that we're going to receive from the environment, and our brain will integrate. So, what is the stimulus? Is it necessary to actually respond? So, what our brain will integrate the stimulus and provide or provide necessary feedback, such as, for example, increasing your temperature, allowing your sweat gland to release sweat, such things like that, like a neural hormone release. So those are the actual response to the stimulus. So we're actually going to dive more into it as we go further. So that is the basic mechanisms. There's a lot of parts to it. We're going to talk more into brain structures, how each structures in regions can contribute to many different things. And we're going to talk about how nerves or fibers perform or structuralize to bring certain feedback and certain stimulus to our brain. So human adult brain actually is almost 97% of the body's nervous tissue. So it's heavily concentrated. So average weight is about 1.4 kilograms, so 3 pounds. So that is the average weight. So the weight can be differ based on the race and ethnicity actually. And the volume is typically about 1,200 milliliters. So your average, it can actually range from 750 to 2,100 ml, but it really differs in the functionally in normal individuals. And males have 10% larger than the females. So there's actually no correlation between brain size and intelligence. So because you have a bigger brain, it doesn't mean you're more smart. Just to know that. So it's better to know the regions of the brain in such ways that you go from the largest region to the smallest region. So we're actually going to I'm actually going to summarize this right now. Our cerebrum is the largest portion of the brain. So a lot of people will say that, cerebrum is the brain, but technically brain refers to everything, including cerebrum and diencephalon, everything. But cerebrum is just part of the brain. And we have diencephalon, which such as thalamus and hypothalamus. And we have brainstem, which is the brain stem of the brain that contains midbrain, pons, and medulla oblongata, which involved in relay centers, relaying sensory information, delivering feedback, and sometimes we have so many different things, reflexes that can happen in medulla and pons. And midbrain actually functions to do a lot of things that can be done. And cerebellum focus more on like movement, coordination of movements. So we're going to dive more into it later. So cerebrum is, like I said, the largest part of the adult brain. Okay? It's part of adult brain, not brain itself. So cerebrum controls higher mental functions. So cerebrum is where your, such as decisions, judgment, consciousness, cognitive, intellectual thinking, critical thinking, thoughts, memory, that's all where it happens. And it is divided into two hemispheres. And such as left and right hemisphere. And we're going to talk about how left and right differs in functions. But right now, all we need to know is that cerebrum is the largest part of the brain and does a lot of higher mental functions. Okay? And we're going to dig more into cerebrum, which is the cerebral cortex. So cortex is a surface layer of gray matter. So brain is composed of two matter. So gray matter and white matter. When we go over spinal cord, we'll see that the difference is the matter. What I'm saying is your brain has gray matter outside and white matter inside, but in spinal cord it's flipped. So we have the white matter on the outside layer, and inside layer we have the gray matter. So those are the just FYI for you guys right now. So we can actually distinguish the structure of the cortex based on how it looks. So rounded elevations, gyri, singular, or we call it gyrus. Okay. So brain, if you unfold the brain, it's actually pretty long. So to actually increase the surface area, and we have to kind of crumble it into one ball, one bit brain, we actually have to crumble it into gyri. So it's to actually increase the surface area. And gyres are separated by two different things. So we could have either sulci or sulcus, which is a shallow depression. And if those sockets are deep enough, we call that a fissure. So fissures are usually what divides the brain regions. And sulci is it's like a smaller version of fissures. So it's also used to divide the brain regions. So this is a cerebrum. We have as you see, the pink region is the cerebral cortex, and we're going to talk more about cerebral cortex, what are they, and what are the components of a cerebral cortex. But just know that brain is technically composed of cerebrum, brain stem, and cerebellum. Those are the three main components, and we're going to dive more into like how each brain stem structure is and cerebellum looks. But know that we have the brain, and we have the gyri, which is the technically, let's think about walking on the canyon. So Grand Canyon. So when you walk on the Grand Canyon, you'll see all the like ridges and shallow grooves, right? So ridges is where you walk on. So that will be the gyri. And sulcus is technically the groove that you're watching all the water flowing. So that is a sulcus. And if your groove is deep enough, you could call that a fissure. So we'll talk about fissure and sulci and gyri more in details later. And we also have the spinal cord that connects with your brain at the level of brainstem. So diencephalon is actually refers to thalamus and hypothalamus. So it's located under cerebrum and cerebellum. Actually, it's not located under cerebellum. It's actually located above cerebellum. So if you look, this is the cerebellum, which is a blue. And if you look into the middle part of the brain, you'll see the red circle and blue. So that is the diencephalon. So technically, I don't know why it says below, but it's technically actually superior above the cerebellum. So it's actually including thalamus, which has a lot of nuclei to process and relay sensory information. So thalamus is like a middleman. It's like an Amazon center. So our sensory receptors will pack and ship. Sensory receptor will receive orders, right? And Amazon is the one that pack and ship it to the location that it needs to go. So thalamus is like an Amazon center. So, and hypothalamus is also a part of diencephalon. It's hypothalamus because right below thalamus. That's it. But hypothalamus is involved in emotions, autonomic functions such as regulating temperature, hunger, satiety, etc., and also releasing hormone production such as creating ADH and many different hormones. So we're going to talk what kind of hormones are released from hypothalamus later. That is the basic structure of cerebrum with cerebellum. And this is diencephalon. So this top structure is the thalamus. And we're going to talk about thalamus more in detail, such as what are the nuclei that actually work together to relay the sensory information. And we're going to talk about hypothalamus, which controls emotions, autonomic function, and hormone production. And also we have pituitary gland, which is the major endocrine gland that actually branches off from the hypothalamus, and it is connected via infundibulum. So you'll see the hypothalamus pointing out towards the anterior side of the brain, and if you look closely, hypothalamus will be connected with infundibulum to the pituitary gland. So this is where your hormones are actually released. So, and pituitary gland actually can be divided into two regions. So we could say the anterior portion, which is the adenohypophysis, and we could separate into posterior region, which is neurohypophysis. So the names are actually based on how it's connected. So adenohypophysis is technically released by adrenal gland. And neurohypophysis is technically named after, it's a direct connection from the hypothalamus. So, like I said, it is connected to hypothalamus via infundibulum, and it can actually integrate nervous and endocrine systems. So we have a hypophyseal portal system, which is like a blood portal system that connects hypothalamus and pituitary gland. These are the region that heavily depends on the blood supply. Okay, and we have the hypothalamo-hypophyseal tract, which is we're connecting from hypothalamus to hypophyseal using the neurons. So we're going to talk more about that. So right here, this is the hypothalamus right here, and that narrow channel right there is the infundibulum. And if you look, we'll have the hypophyseal fossa of the skull, and your pituitary gland was sitting there. And like I said, pituitary gland actually can be divided into anterior lobe and posterior lobe, and depending on depending on the lobe, different kind of hormones can be released. We can actually branch anterior pituitary lobe into three parts. We could have pars tuberalis, which is the most superior portion of the gland, and we have the pars distalis, actually, which is the most distal portion of the gland, and with the pars intermedia is where your it's a location where your posterior and anterior gland actually meets together. And we also have an important structure called mammary body, which will actually play a role for hungers and satiety. And so this is the hypophyseal portal. So this is where your blood supplies work. And this is how your neurons, hypothalamus, secretes hormones. So these are the neurons of the hypothalamus. So we have neurosecretory neurons, which will secrete hormones, and we have paraventricular nuclei, which will actually work. We'll talk about it more later, and supraoptic nuclei, which actually also works on hormones release. So this is the posterior aspect of the lobe, posterior aspect of the gland, and this is the anterior aspect of the gland. So if you see, we have a lot of endocrine cells, which releases hormones. So we could say that adenohypophysis, or anterior lobe of the pituitary gland, is actually independent of a neuron. So it will actually release the hormones via endocrine cells, via exocytosis to your blood vessels, the veins, and those veins will actually carry those hormones to the target cells. And we have the superior hypophyseal artery delivers blood to the capillary network. So this is the superior hypophyseal artery. Just letting you know, artery is not a part of portal. Portal is the connection between the hypothalamus and the gland. So artery is technically not a part of the portal, but allows a blood supply. So superior hypophyseal artery delivers blood to capillary network. So this is the capillary network of the upper infundibulum. So this is where your oxygen will be carried, and the portal vessels deliver blood containing regulatory hormones. So this is the portal vessels. So like I said, artery is not part of the portal because it's not really direct connection between infundibulum and hypothalamus, or gland and hypothalamus. So portal is one that directly connects the hypothalamus and the gland, but artery doesn't. So just make sure you remember that. And the portal vessels will deliver blood containing regulatory hormones, and those will bind to the receptors in endocrine cells, and those endocrine cells will release hormones that will actually diffuse to the vein and carry to the rest of the body. And what about posterior side? So what happened is we have several neurons that actually release neuron hormones to the vessels. So these neuron hormones actually will travel down, so you'll fire axial potential there, and there's synaptic cleft between the vessels, and these neurons will release neuron hormones, also called neurotransmitters, but in this case, it's hormones. It will be traveling to the bloodstream. So these will be released, and our veins will carry those blood that contains neural hormones to to many different target cells. So the major point of this slide is that artery is not part of a portal. Portal is a connection between your hypothalamus and your gland. And based on the lobe, anterior, posterior, it has a different mechanisms. So anterior will actually use endocrine cells, which will release the hormones to the bloodstream. And for posterior, we'll have the neuron directly innervates that vessels and release hormones. And those hormones will be carried with the blood to the target cells. So brain stems. So now that we talked about diencephalon, we can talk about brain stems. It includes midbrains and pons and medulla oblongata. So we're going to talk more about them in later. And the major function is, we have a lot of tracts. We have a lot of fibers that pass through your neck, in your body, in your head, in your brain. So that is the main. So I'll think of brainstem as a major highway system. So where your cars going in and out, all happening all the time, right? So I'll think of brainstem as a highway. Okay, so relays information between spinal cord and cerebrum or cerebellum. So spinal cord. So that'll be CNS. So brainstem is technically the connection between the CNS, spinal cord, and brain. And it includes midbrain, which is the upper portion of the brainstem. We'll talk more about how it looks like. And we have the pons and medulla oblongata. So midbrain does really important things. So it processes sight, sound, associated reflexes, and also maintain consciousness. So when we talk about consciousness, we could actually talk about consciousness as the cognitive levels or consciousness as your physical levels. So when you're alive, right, that's consciousness. You're conscious, right? So, but if you talk about the cognitive level, consciousness actually can be dived into more like judgment, decision making, right? So we can actually separate into two. So the consciousness in this midbrain is what I'm talking about is whether you're alive or not. Okay, so it maintains consciousness, and pons actually connect cerebellum to brain stem. So such as we have peduncles, cerebellar peduncles that will connect pons and cerebellum, and we'll have a lot of tracts that passes through cerebellum and pons and midbrain that will actually allow coordination of movement and many different things. It also contains tracts, collection of CNS axons, and we're going to talk more about it. And we have the medulla oblongata, which connects brain to the spinal cord. So we'll have a lot of tracts that will pass through. And it's the inferior portion and has a central canal. So central canal is where your CSF will lastly flow through. So your CSF will actually flow around your head, and the final destination will be the central canal, and it will flow all the way down to the spinal cord and back to the brain and released via arachnoid granulation. Okay, so relay sensory information. We have a lot of nucleus that involved in visions, auditory, and vestibular. So we're going to talk more about that. It also regulates heart rate, blood pressure, and digestion. So that's what we just talked about: cerebrum, cerebellum, and brainstem and diencephalons. Right below the thalamus, you'll see the midbrain. Just make sure don't get confused which side is the front. Okay, so you'll see the peduncles on the posterior side. So this is the dorsal aspect of the brainstem. So posterior, and so you can't say posterior anterior anymore. So this will be dorsal aspect of the brainstem. So when you talk about brainstem itself, you can't just say posterior because posterior means back. So we have to use terms ventral and dorsal. So this will be the dorsal aspect of the midbrain. No, dorsal aspect of the brainstem. There you go. Because you'll see the peduncles that will connect the there's midbrain. Yes, there's midbrain and the pons, right, and the medulla. How do I know which way is dorsal and which way is ventral? So when I look at the pons, pons usually have peduncles that connect cerebellum on the dorsal aspect. So you'll see three peduncles: superior, inferior, middle. And if you see those peduncles, that's the dorsal. So your brain will actually have structure like a 7, right? So, and then how do I know? And you can also look at the midbrain to figure out which way is dorsal and ventral. And so if you look, midbrain will have these olive nucleus right here. So these are the olive nucleus. So if you see the olive nucleus, that's definitely dorsal aspect, and you'll see you'll see the pyramid of the medulla too. So that's also dorsal side. So cerebellum is the second largest of the brain. It's like a mini brain. So what it does is, to simplify, what it does is actually coordinates repetitive body movements. So when we initiate the movements, right, and initiating movement is not that hard, but regulating and fixing the errors while during the movement is hard. And cerebellum does that. So cerebellum, our primary motor cortex will actually initiate the movement, right? Move your muscles. And while movement is going, your cerebellum will actually receive a lot of stimulus based on your muscle position, how your muscles moved, and if there's any errors in that movement, your cerebellum will fix it, make it right, and send it back to your brain, and your brain will innervate those muscles. So that's how it works usually. And I also have two hemispheres, just like cerebrum. And it's also covered by gray matter. In this case, we call that cerebellar cortex, and we're going to talk more about it later. I'm going to go and skip that. Okay, so how does our brain forms? Okay, very important. So this is during embryonic development. So we have neural plate, which after the zygomatic, after proliferation, your zygomatic starts dividing, it'll form a shape into a neural tube. Okay, so those neural tube will develop to the CNS, and the rostral portion, which is the kind of similar to the anterior portion, but rostral is a little bit different. But rostral portion actually enlarges into three primary brain vesicles. Those are the three primary initial brain vesicles, which is called procephalons. Prosencephalon, which is forebrain, and we have mesencephalon, which is the midbrain. My quick tip for you is that mesencephalon will always stay mesencephalon, the midbrain. So that's the only one that actually doesn't change. Okay, and the rhombencephalon is the hindbrain, which will further divide into mesencephalon and myelencephalon. So quick way to remember is you have tell die and I'll meet my. So that's one way to remember. So tell die until I die, I will meet my. I don't know if that's gonna help, but that's how I remembered back in neuroanatomy class. But it really did help me. But I'm gonna go over this one more time. So primary brain vesicles can be divided into prosencephalon, which is the forebrain, mesencephalon, which is the midbrain, which will stay as mesencephalon, and rhombencephalon, which is the hindbrain, which will further divide into metencephalons and myelencephalon. Okay, and we're going to talk more about those secondary brain vesicles now. There you go. So when these primary brain vesicle will further divide, it will divide into secondary, like we just talked about. So telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. So quick overview. Telencephalon would divide telencephalon would be your cerebral cortex and the lateral ventricles. Yes, cortex and the lateral ventricles. Your diencephalon will be the diencephalon, which will be thalamus and hypothalamus, and also form ventricles. Third ventricles. Okay, third ventricles. Yes. And mesencephalon will actually stay as midbrain, and metencephalon, what's below midbrain? Pons and cerebellum. So metencephalon will actually become the pons, cerebellum, and cerebral aqueduct, which connects your third and fourth ventricles. And myelencephalon, right? Myelencephalon will become the medulla oblongata and the lower fourth ventricles. So going over again, telencephalon will form cerebrum and lateral ventricles, and diencephalon will remain as diencephalon in third ventricles, which can be divided into thalamus and hypothalamus, and mesencephalon, which is it's gonna stay as midbrain, and formation of cerebral aqueduct, which connects fourth and third ventricles, and metencephalon will form cerebellum, pons, and upper fourth ventricles, and myelencephalon will form medulla oblongata and lower fourth ventricles. So these are very important to memorize. And next slide will show everything. There you go. This is the neural after your neural tube will fold. Neural plate will fold into two, and it will actually divide into three primary vesicles, which is prosencephalon, the forebrain, midbrain, mesencephalon, and the hindbrain, the rhombencephalon. And after about 6 weeks, these primary brain vesicle will further divide into secondary brain vesicle, which now we call telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. So telencephalon will become cerebrum. And what kind of ventricles? Lateral ventricles. So diencephalon will become diencephalon, which is thalamus and hypothalamus. What kind of ventricles? The third ventricles. And mesencephalon will become the midbrain, which will become the cerebral aqueduct that connects third and the fourth. Metencephalon will become the cerebellum and pons, which will become the fourth ventricles, the upper portion. And the myelencephalon will become the medulla oblongata, which will become the fourth ventricle, the lower portion of the fourth ventricles. All right. Why is ventricle important? To summarize, whatever is going to be on the next slides, to summarize why ventricle is important is to actually cushion your brain. Your brain needs to float. Your brain just cannot sit in your skull like dangling around when you walk and run. It cannot just be like that. It has to be floated and cushioned, right? So that's the primary function of ventricles. Okay, so in embryo, neural tube encloses neural coil. Okay, neural coil expands to form chambers and ventricles lined with ependymal cells. Glial cells, which will produce CSF and line the ventricles, right? We learned this from chapter 12. Very important. And each cerebral hemisphere contains one large lateral ventricles. Each cerebral hemisphere contains one large ventricles. One more time. Each cerebral hemisphere, so left and right hemisphere, contain one large lateral ventricles. So one left lateral and one right lateral ventricles. And they're usually separated by septum pellucidum. Okay? So you notice we don't have second ventricles, okay? In your exam, don't get confused with, hey, which of the following will derive from diencephalon? And the answer's choice is something like primary ventricles, lateral ventricles, secondary ventricles. Okay? There's no such primary vesicles and the ventricles, or there's no such secondary ventricles, second ventricles, whatever. Just make sure there's lateral ventricles towards the lateral side of each hemisphere, and we have the third ventricles, which is located near the hypothalamus and thalamus, the diencephalon, and near the brainstem, such as pons and your medulla and midbrain, will have the fourth ventricles connected with cerebral aqueduct to third ventricles. Okay. So like I said, lateral ventricle separated by septum pellucidum, and we have the third ventricle and diencephalon, and actually they communicate with each lateral ventricles via interventricular foramen, which is called foramen Monro. So you'll have third ventricle that will connect it to the lateral ventricles via interventricular foramen, and we're also named foramen Monro. So fourth ventricles extends into medulla. So this is where your fourth ventricles will extend into medulla, become central canal. Okay. So it joins central canal of the spinal cord and connects with the third ventricles via cerebral aqueduct. So this is the actual ventricular system of the brain. As we can see the lateral ventricles towards the lateral side of the two hemispheres. So we have left and right. And right there, we have the interventricular foramen, which connects the third ventricles to the each lateral ventricles. And we have the cerebral aqueduct, and we have the fourth ventricles. Okay, so let me tell you the direction of the CSF flow. Okay, so it will flow from ependymal cells. Actually, let's go over those first. This is the anterior view, so you'll see the lateral horns. So this is the lateral horns of the lateral ventricles, so you don't have to know those. But this is the lateral ventricles and third ventricles right there, connected to interventricular foramen. And you'll see the fourth ventricles connected to third ventricles via cerebral aqueduct, and those fourth ventricle will become central canal in the spinal cord. Okay. So, CSF also does that. But what else protects our brain? Our skulls. Right? We already learned the cranium, so we don't need to learn those again. But there's another one that is very important, which is the cranial meninges. So meninges is the protection layer that protects your brain. It's composed of 3 layers. So the most superior upper layer is the dura mater. And right below we have the arachnoid mater, and our thinnest layer that is with contact with the brain is the pia mater. Let me tell you, dura mater is very hard, and meningitis, which is the bacterial infection on your meninges, actually happens in where? Arachnoid and pia mater. So dura mater is actually pretty thick enough, so it's really hard to get the bacterial infection in there. So what actually happens is your arachnoid and pia mater is the one that get always the bacterial infection. Okay, we're going to talk more about it. And we also have blood-brain barrier formed by astrocytes, which filters out the blood. Actually does a lot of things like diffuse in your blood and filter, such as using different chemicals, and filter that blood back out to the brain so that your neurons can actually stay in healthy environment. So we have, like I said, we have 3 layers for the cranial meninges. We have dura mater, arachnoid, and pia mater, and they're actually continuous spinal meninges. So those meninges will actually continue all the way down to the spinal cord and protect the spinal cord too. All right. Okay, so we're going to talk about the dura mater. So that's the most superior layer of the meninges, which is the inner fibrous layer. We have the meningeal cranial dura, and we have the outer fibrous layer, which is the periosteal cranial dura. So, based on the name, outer fibrous layer will connect to your periosteum, which is your skull. And we have the inner fibrous layer, meningeal cranial dura, will be contact with arachnoid mater. Okay? So that's the cranium. We'll have dura mater that's in blue, and we have the periosteal cranial dura contact with the cranium, and we have the meningeal cranial dura, which contact with the arachnoid mater. And of course, we do have space. We do have potential space. So I said potential, all right? Potential means it could be used or not. So that could be a potential space. That is called dural sinus. So that's a sinus. So this is where CSF will actually flow around. Okay, so we want to talk more about that later. But major layer of the dura is the periosteal cranial dura, and then meningeal cranial dura, which contact with arachnoid mater. And between we have the dural sinus. And we're going to talk about dural folds. So these dura mater actually will make a fold. Oh, surprising. Why? Well, I don't know if you went over earlier. I said that our brain can be divided with fissures. So your brain can be divided into regions, or structures, or regions. Yes, regions. So when there's like really deep fissure, right? There's gonna be space there, alright? But how is your dural matter actually gonna protect all that layers? Well, so your dural matter is actually gonna fold into it. So it will actually fold and all the way to the fissure and protect the brain. So it's called dural folds. So we have three largest dural folds, which is called we have falx cerebri. Okay, falx. Falx mean cutting through. So you're cutting through cerebri. So this will actually align with the longitudinal fissure. So when you're dividing the brain to left and right, that line, that fissure, your falx cerebri will line up that fissure. And we have the tentorium cerebelli, which will actually line up the protect the cerebellum. And we have falx cerebelli, which will actually divide your cerebrum with cerebellum. Let's look at it. All right. Falx cerebri projects between cerebral hemisphere. Like I said, it contains superior. It's gonna divide. That longitudinal fissure is gonna fold into that fissure to separate the left and right hemisphere. It contains superior sagittal sinus and inferior sagittal sinus. So your CSF will actually either flow from inferior or superior sagittal sinus. And when those sinus will move around the brain, going towards the superior and travel, what happens is you'll meet tentorium cerebelli, which separates cerebrum from cerebellum. So that line, that fissure that divides them, you will have tentorium cerebelli, which contains transverse sinus and straight sinus. So your CSF will flow from inferior and superior sagittal sinus. They'll become one sagittal sinus, and eventually they'll become transverse sinus, and either they could either go to straight sinus or different or occipital sinus. And we also have falx cerebelli, which divides cerebellum hemisphere, right? So it also contains occipital sinus. So I think it's better to go over the pictures for this. So okay, there you go. Okay, so the one that cuts through the hemisphere is called the falx cerebri. This is the inferior sagittal sinus. Sagittal sinus because it's cut through the sagittal way. So that's what sagittal sinus. So this is the inferior sagittal sinus. And above that, we have the superior sagittal sinus. So do you guys see how it works? Yes, you'll see. Because your CSF actually can flow from this way, from superior sagittal sinus all the way to transverse sinus, or it can flow from inferior sagittal sinus all the way to transverse sinus. This right here is called straight sinus because it connects your inferior sagittal sinus to your transverse sinus. So that's the straight sinus. So like I said, we have the falx cerebri separating the two hemisphere, and we have the tentorium cerebelli, which separates your cerebral cortex or cerebrum from cerebellum. And we have the falx cerebelli, which divides your cerebellar hemisphere. And we have several different sinuses. We have the inferior sagittal sinus, and with the superior sagittal sinus. And eventually, inferior sagittal sinus will connect with straight sinus to meet at the transverse sinus with sagittal sinus, superior sagittal sinus. So that's the straight. And it will eventually pass through your other sinuses and exit through. This is how it works right here. So we have the superior sagittal sinus, the most superior one, and we have the inferior sagittal sinus, and we have the straight sinus, which connects your so that connection is called confluence of sinuses. So that right here, that connection right here is called the confluence of sinuses. This is where your inferior sagittal sinus, your straight sinuses, superior sagittal sinus, and transverse sinus meet together. So what happens is your CSF will flow from superior sagittal sinus to your confluence of sinus. Your inferior sagittal sinus will flow through straight and two components of sinuses, and those will actually then travel to the transverse sinus, and eventually they'll exit as an internal jugular and vertebral vein. So now let's talk about arachnoid matter, so which covers the brain. So so arachnoids is right below dura, which covers the brain. So we have the arachnoid right below the dura, and dura actually has those subarachnoid space, which is like let me show find it. Yeah, let's use this. So this is the arachnoid matter. But if you see, there's like a spider webs, right? Those are called um, okay, those are called arachnoid branches. Okay, and these branches will actually like form like cushion wise, and these empty spaces or potential spaces called subarachnoid space. So this is where your CSF will actually make and first float around and to the sagittal sinus and to sinuses. So I'm gonna actually talk more about the flow of CSF later, so just make sure you tune on, okay? But just know that this is the subarachnoid space, and we have the arachnoid matter connected to the dura, and subarachnoid space, and we have the pia matter. And CSF surrounds an exposed surface of the central nervous system, and the functions to support the brain, yeah, makes the brain float, and cushion delicate neural structures, cushions, and transport nutrients and chemical messengers and waste, right? Okay, there you go. That is beautiful. Okay, all right. So, the first place we need to start is we need to make the CSF. So, where we make the CSF? Choroid plexus of third ventricle. So, your third ventricle is where a massive amount of CSF is produced. So, this is where your choroid plexus is located. So, it actually contains a lot of ependymal cells. So, these will actually line up the third ventricles, and choroid plexus of third ventricle, which composed of ependymal cells, they will secrete CSF. Now, the secreted CSF will actually flow through the choroid plexus through the ventricles and towards the fourth ventricles, and fills in the central canal and will flow down to the spinal cord. Okay, and at the end of the spinal cord, what happens is you'll have what end of the spinal cord will actually flow back up. So they'll actually flow through the subarachnoid space and go all the way back up to the to the brain. So this is subarachnoid space. So actually pass through all the subarachnoid space, and it's gonna float around until you get to the arachnoid granulation. So this is where your arachnoid matter penetrates through the dura matter. So you have the dura matter, which is the meningeal dura, right? So that arachnoid subarachnoid space or arachnoid matter will actually penetrate through that meningeal dura matter, and this is where CSF is released. And once your CSF is released, it can either pass through the superior sagittal sinus or inferior sagittal sinus, and then cycle continues from there. And after that, once it reaches transverse sinuses, and it will actually go down to the jugular vein and vertebral vein. All right, going over again. So we have the third ventricle, which contains choroid plexus, has a lot of ependymal cells, which will secrete CSF. And those CSF will actually travel down through the ventricles, cerebral aqueduct, fourth ventricles, and to the central canal. And at the end of the spinal cord, it will actually flow out through the subarachnoid space, and then travel around the subarachnoid space until we get to the arachnoid granulation. And granulation is the extension of arachnoid matter through the dural matter, and our CSF is released, and those released CSF can actually travel through the sagittal sinuses and all the way to the transverse sinuses. And from the transverse sinuses, those CSF are released from the jugular towards the jugular veins or to vertebral veins. All right. Why is choroid plexus important? Because, like I said, they're the one that produce CSF. So they have a lot of ependymal cells around the capillaries. So every day it's about 500 ml of the CSF into ventricles per day, and the primary function is to make, of course, produce CSF and remove waste from products from CSF and adjust composition of PSF. So like viscosity of the CSF, how fluidy, right? Okay, so this right here is a choroid plexus lined up with ependymal cells, and what happens is your CSF will actually diffuse through, right? And then your ependymal cells will actually filter out and release back. And those CSF will actually flow through the subarachnoid space to the sinuses and to the sinuses and back to the vein. So we have CSF circulates from choroid plexus to the ventricles and central canal to the spinal cord, and it's going to actually into the subarachnoid space to lateral apertures and medial aperture, and to surround the brain, the spinal cord, and cauda equina. And after that, it'll flow out subarachnoid, yes, and we have the arachnoid villi extension of arachnoid membrane. That's the arachnoid villi, and which will form arachnoid granulation. A large cluster of arachnoid villi in adults. This is where you'll absorb CSF into venous circulation. So after it flows through subarachnoid space, your CSF will flow through the arachnoid granulation, which contains a lot of arachnoid villi, and it will continue their way through the sinuses. Okay, all right. Yes, this is what I was talking about. So it actually penetrates through the dura mater. So this is the meningeal layer of the dura mater, and you'll see that arachnoid matter actually penetrates through, so that your CSF can be actually released to the sinuses. And we have a lot of arachnoid villi, which will form this cluster called arachnoid granulation. So if you have any questions, just leave a comment. If I can I'll try my best to actually reply. If there's a conceptuals, just leave a comment, and I'll try my best to answer your comments. Maybe not. I'm probably busy too, so I'm trying to study for the exams too. So just let me know. Okay. All right. So what happens when we damage the folds or the meninges? So we can ask several things. So mostly what happens is dura mater is the thickest layer of the brain. So what we'll do is okay. So let's think about a case. I hit my head in a wall. My skull's cracked and penetrated through the dura mater, and it started bleeding. So depending on how your blood is clustered, we can actually determine whether the blood is from the subdural space or actually from epidural space. So like I said, dura mater actually has two layers, inner and outer, which connects your cranium and your actual arachnoid matter. Between, we have the sinuses, and also between the connection between your periosteum and the periosteal dura mater. There's space actually, so it's not really tightly connected, but there's potential space. It's called the epidural space. What about subdural space? Well, it's your dura mater will be connected to your arachnoid mater. Right below that is called a subdural space, right? So it's not actually tightly connected. It is tightly connected, but we just signify the potential space between those connections. So, like I said, epidural is between the cranium and the a periosteal dura mater layer, and subdural should be the potential space between arachnoid matter and meningeal dura mater. Alright, so two possibilities. We could have a bleeding in the epidural space, or we could have bleeding in the subdural space. What is the difference? Well, bleeding in the epidural potential space, or we could have bleeding in the subdural space. So depending on how severe your trauma is, cranial trauma, we could either have both, right? Actually, we can identify the hematoma, which is the bleeding of the cranial, based on shape. So this is what you will learn in neuroanatomy if you're taking neuroanatomy class. Based on your bleeding shape, you can actually determine whether the bleeding is subdural hematoma or epidural hematoma. So epidural hematoma actually shaped like a lemon under this MR scan. So under the MR scan, it looks like a lemon shape. And if you look at the subdural hematoma, it actually looks like a banana shape. So that's how we distinguish the hematomas, okay? But that's not really important for this lecture, so you guys don't have to worry about it. But just know that cranial trauma actually can allow bleeding in your brain. It can actually damage the meninges that protects the brain. So we have several blood supply to the brain. So to begin with, our heart has a lot of vessels that comes out, right? So